Good afternoon and welcome to this PDBE webinar. My name is David Armstrong. I'm part of the PDBE annotation team. So we deal with the deposited structural data for both the Protein Data Bank and the Electron Microscopy Data Bank. And this is a webinar from the EMBL-EBI webinar series. So the PDB and the EMDB, the Protein Data Bank and the Electron Microscopy Data Bank, are archives, and along with our partners in the Worldwide Protein Data Bank, us at the PDBE are responsible for the collection and the distribution of these archives. And these specifically are for experimentally determined macromolecular structures. So to begin with, I'll tell you a bit about our homepage, how you navigate the homepage. I'll also tell you how you would deposit a structure if you have one to deposit to the databases. I'll then move on to talking about the search. We have one search bar to keep things simple. There are also facets to enable you to narrow down your search. And we also offer you some different views of your search results. I'll move on then to talk about entry pages. You can find on these entry pages information about the citations involved with that entry, information about the molecules in an entry, also information about the ligands and validation information as well. We also have a number of tools that we've developed here at the PDBE. These include PDBEFOLD, which is used for searching for structural similarity. So much like BLAST is for sequence similarity, this uses structural elements to find proteins that are similar to yours of interest. We also have PDBE PISA, which is used for crystallographic entries, and it calculates the likelihood of the interfaces within a crystal occurring within solution. We also have an API which is used for more programmatical and bioinformatics information. I'll also tell you about the Electron Microscopy Data Bank, how you would search this database for electron microscopy volume data, different ways you can view the data using our volume viewer and image viewers, and finally EMPIAR, which is the Electron Microscopy Pilot Image Archive, and I'll introduce that. Our homepage at pdbe.org is your starting point for information on macromolecular structures. So I'll show you how to get there and what you can find on the homepage. So to begin with, you can type pdbe.org into your browser, and that will take you directly to our homepage. There are some tabs at the top of the page here to navigate. If you want to deposit structures, you click on deposition. There are a number of services you can also navigate to. We have a training tab for tutorials and more information about our services, and a couple of other tabs regarding more about us. We also have a featured structure section, in this case the Zika virus, recently solved. There's also links to popular services on our website. For those interested in the EMDB, there's a link to the search. There's the link here to PDBEFOLD for searching structural similarity, one for PDBE PISA for searching crystallographic interfaces, our API for programmatical resources, and also a link to EMPIAR, our Electron Microscopy Pilot Image Archive. We have our latest archive statistics here, and the database is updated each week on a Wednesday, and you can access the latest entries from those links. We're also active on social media. We have a YouTube channel that you can go to, and this webinar will be made available on there. There are also Facebook and Twitter accounts, so you can like or follow us on there, and our tweets are embedded on our webpage. We also have our events shown here if you want to see what we're up to, where you can find us, and also news of the latest structural biology information and what's new in PDBE. If you're looking to deposit a macromolecular structure, then this can be easily found on our website. Again, you go to pdbe.org and click on the Deposition tab at the top of the page. This takes you through to our deposition services. And as you can see, the two links at the top have new next to them. That's because we have a new deposition system, which is now common to both PDB for the Protein Data Bank and for the EMDB, the Electron Microscopy Data Bank. These are all now dealt with a common system. Further down the page, there's a link for validation, so you can use our standalone validation server to validate your structure before you deposit. If you click on one of these links at the top, it will take you to our deposition system. You'll see at the top left that it's the worldwide PDB. That's because this is in collaboration with our colleagues at RCSB and PDBJ, as well as BMRB, who deal with NMR data. So the first thing you would do is select your country, where you are depositing the data from. This because your data is dealt with by geography. So we at the PDBE would deal with the European entries. You can then select your experimental method, and there are also a selection of accession codes you can request depending on what you're depositing. So a structural model for the protein data bank would require a PDB accession code. However, for an EM entry, you may also want to do your map submission in one entry, in one deposition. So you can also select to request an Electron Microscopy Data Bank accession code as well. There is also a BMRB link there if you are depositing NMR data. Searching at the PDBE is simple. We make it easy for you to quickly find what you're looking for. So I'll show you how we do this from our web page. If you go to PDBE.org, you'll see at the top right of your page that there is one single search bar. So rather than having to fill in a lot of advanced search forms, you can just click in the single web page, the single search bar at the top. There's also a link here for the EM search. So if you're looking for electron microscopy map data, you can search from there. If you begin typing in our search bar, you'll see that these autocompletes arrive below you, and you have a number of different sections here to narrow down your search further. If you're looking to select by a particular enzyme category, you can see that the numbers here are from biggest to smallest. That's because the most populous of these categories are given at the top. So in this case, hydrolases are the most common enzyme category with hydro in the name. We also have a number of gene ontology annotations thanks to our links to UniProt, such as components, function, and processes, and you can search these for a protein of interest based on these. If you want to search by journal, any entry in this case with hydro in the journal title will come up, and in this case only the Journal of Hydrogen Energy. There's also a selection here for ligands, in this case with hydro again in the ligand name. If you want to be more specific in your molecule search and search for molecule name, this is also an option. And you can also search by organism, and again, organisms with hydro within the name will come up in your search. When you're happy with what you want to search for instance, hydrolases, you can then click on that, and that will narrow your search based on this facet. Facets in our search pages make it easy for you to narrow down your search. So here is a search for hydrolases, and you'll see at the top of the page that hydrolases is shown, the facet that we've chosen already. However, there's still a large number of entries in the search, so we want to narrow this down further to find entries specific for what we're looking for. You'll see on the left that there's a number of facets that you can select from macromolecules, there's molecule type for proteins and nucleic acid. There's also species name. For instance, if you wanted to look for only human entries, human macromolecules, you can click on Homo sapiens, and you'll see at the top of this page, the facet is now shown at the top. And these are now the human hydrolases. If you want to narrow down your search further, you might select another facet. For instance, if you looked at assembly polymer count, this is the annotated assembly. And maybe you want to narrow your search by dimer. You would select this, and this will update your search based on dimers. If you decide you want to remove one of the facets, you can easily click the cross next to hydrolases, and this will be removed from the search, narrowing it down further. At the PDBE, we're moving away from the more entry-centric search results. I'll show you how you can view your search differently here. So above the search results, you have these 4 tabs for entries, macromolecules, compounds, and protein families. I'll begin to talk firstly about our entries view. You can see here they're listed by individual ID code, individual entry, in this case entry 2C6G. You have some information shown initially on the publication details. the organism and assembly, and also some interacting ligands. The experimental type is given in the top right, along with the resolution if applicable. And you also have validation sliders, which give you an idea of how many outliers might be in the entry. There's also images here to give you an idea of what the best assembly will look like. And we give that in 3 views for an easy view, for an easy snapshot of the structure. You can also sort your results, for instance, by quality. And this will give, at the top of the page, the best validation score, that is the ones with the least outliers. If you click on the macromolecules tab, instead of sorting by entries, things are sorted by individual macromolecule. For instance, this is adenosine deamidase. In the image on the right, it will show where this individual macromolecule is within that entry. Some macromolecules will have multiple entries. We give you the best example based on the validation score, and we also give you the other entries, and these are ordered by best validation score to lowest, i.e. least outliers to more significant outliers. There's also a compounds tab, and this lists things by individual ligand. For instance, this is ascorbic acid. Again, we give a best example as well as other examples. If you hover over the name, you can see that the chemical structure comes up for that ligand. There's also more images shown on the right here. If you click on that, this opens the box, and we show a ligand view where it is within the structure, shown in blue, and also a ligand environment. The protein families tab separates entries by protein family rather than by entry type, and again gives best example and where it also occurs. You also get an image on the right here of where that protein family domain is within the structure. Again, before we move on, I'd just like to give another poll, if you wouldn't mind filling that out for me. So the majority of you said X-ray crystallography, which is correct. There are, in fact, the vast majority of PDB entries are X-ray entries. A few people said homology modeling, but we actually don't accept homology models. We only accept experimental data in the PDB. And electron microscopy, no one did go for. It's a relatively minor group within the PDB, but it is a very fast expanding field. So each entry page in the PDBE describes a single experimental data set. I'll show you what an entry page looks like here. So this is entry 3DDF, and you'll see that you get some information about the experimental technique. You also get an image box here that you can click on to expand. This gives you a number of different images based on a number of different views, so you can see what the enzyme, the protein or nucleic acid looks from different angles. You also can view where individual macromolecules are within the entry, and also an assembly view, which shows you the annotated assembly for that entry. We also give citation information on the page. You'll see in this box, this gives the publication details for that entry. There's also details for where it is cited. This entry has been cited in other papers. The function and biology section, thanks to our links with Uniprot and PFAM, gives you more information about the biology. There's also a Ligands and Environments section that lists all the ligands within this entry. Our experiments and validation section gives you a chance to see how many likely outliers there are in an entry. And finally, our structure analysis section, again thanks to links with Uniprot, gives you more information about the individual molecules in an entry. Citation information is easy to access on our entry pages, and I'll show you how to do this. So this entry is 2BPX, and you'll see at the top left here in the grey box you have some information about the publication for this entry. There's also a PubMed ID link. This takes us to our colleagues at Europe PMC, PubMed Central. There's also a link on the right here to our main citations page. This gives you even more information about the publication itself. You also get to see how many times the entry has been cited, and any entries related to this one, if you're looking for similar entries. There's a DOI link here, which links straight out to the paper, so that you can view the full paper if you want to. We, however, also give you an abstract here, so that you can get an idea of what is in the paper without having to find the paper itself. This section here for articles where the ID code is mentioned but not cited, so if someone has referred to the structure in a paper but haven't cited it, we will still pick this up thanks to EuroPMC's data mining. We also show articles that cite the publication, including, if applicable, reviews, and finally any related citations that the authors have provided. For open access journals, we also can find figures within the paper. You'll see this image box here that you can expand. Along the bottom you have thumbnails for each of the figures within the paper, and you get an expanded image along with a figure legend for each of these figures. And you can easily navigate through these one by one to see all the information within the paper. Our structure analysis section gives you information about the individual macromolecules within an entry. If you go to an entry page, for instance, this is a hemoglobin entry, 5E6E, scroll down to the structure analysis section, you can see there's multiple molecules within this. If we click on the molecule details for the hemoglobin subunit beta, you find out more information about this individual macromolecule. So there's some more information at the top there, including a sequence for if you want to do blast searching. The image will give you where the molecule is within the structure, highlighted in blue. If you go further down the page, you'll see our visualization tool. So we have this in both 1D, 2D, and 3D views. On the 1D view at the top, if you navigate to the molecule section, these parts above the molecule tab show you where any conflicts or engineered mutations might be. There's also a UniProt link, and you can see here which sections of the protein corresponds to the UniProt range, and this is highlighted on the 2D image as well. There's also a PFAM link here. Again, this connects to the 2D image. If you click on this, this will also open this within the 3D viewer. So these are all inherently linked. On the 1D chain viewer, this maps also to the 2D viewer below and vice versa. It also gives you any residues which are within the binding site of ligands. So you can see this within the sequence. The quality section points out any validation issues you might find, some more minor issues in yellow and more major issues given in red. There's also a section for secondary structure. This is shown on the 2D, and again, if you click on this, this will be highlighted in the 3D image. Another tool within the 2D section is the ability to color by quality, and this indicates where on the 2D image any issues with the validation might be. Again, you can click on these and view these peptides in the 3D viewer. Many ligands, for instance drug compounds, are often actually the most interesting part of an entry. So I'll show you how to navigate to the Ligands and Environments section on our webpage. So for an entry, for instance 5F74, you can see here there's a Ligands and Environments section, and any ligands within this entry will be listed here. There's also a link to Ligands and Environments section on the right. Again, all the ligands shown here are in a 3D view in this case, so you're able to view the environment here without downloading the structure. If you want more details, you can go to the environment details link here, and this takes you to the page specific for that ligand in this entry. So this example is adenosine monophosphate. You get some information at the top here, as well as an image of the structure of the ligand. There's also an image of where this ligand is within the structure, highlighted in blue. Further down the page, we give more information about the environment details for the ligand. This is shown on the left in 2D, and you can see where the ligand is bound, and what is associated with the other molecules in the entry. You can also view this in 3D, so a larger version of the previous screen, and you can view the environment easily here. Coming soon is our new ligand viewer. This will include the addition of electron density, so we'll now be showing you the data as well as the model. And we can also show difference maps, and this indicates where data might be missing from the model or where X data has not been modeled. As part of the worldwide PDB, the PDBE has been involved in developing the validation of PDB entries. So I'll show you the experiments and validations section. We have a quick view here of the validation sliders. This is shown in more detail in the experiments and validations section down the page. So this includes things like R3 and Ramachandran outliers and sidechain outliers. The further to the right, in the blue these bars are, the less outliers. The further to the red, the more significant the outliers in this entry. You can click on further details, and this takes you to the main page for experiments and validation. Again, you have the image shown at the top. You can also scroll down for more validation information and also some experimental information. Finally, you can see a full validation report. This is what we now recommend is sent with the publication of each structure. There are a number of tools that we've developed here at the PDBE that you may find that are useful to your own research. PDBEFOLD, for instance, helps you to find proteins based on their structural similarity to a protein of interest. We also have PDBE-PISA, and this is used for calculation of the interfaces between molecules in a crystal entry and suggests which of these are likely to occur in solution. We also have our REST API, and this is used for programmatical access for data, so people interested in bioinformatics or large data sets. I don't have time to go into too much detail, unfortunately, in these, but I'll let you know how to find these services and find more information about these. So if you go to our homepage at pdbe.org, at the top right here, you'll see linked here for PDBEFOLD, and this will take you to our Fold server. There is a launch button here to launch the service, and you can find any structurally similar proteins to yours of interest. You've then got the ability to view the superimposed structures and see where these similarities are on the structure, and maybe where any differences are as well within these structures. Another of our tools is PDBE-PISA, and you'll find the link here on the top right, which will take you to the PISA server. Again, from this page, you would launch the service, and there's a launch button here. And PDBE-PISA will find likely assemblies based on crystallographic interfaces. This is also found in the Structure Analysis tab of our entry pages. As you scroll down the page, there's a section for assemblies here, and this gives you information about the surface area accessible and buried, the multimeric state, energies associated with the formation of the assembly, and also images, so that you can see what the entry looks like as an assembly. Finally, you can also download these assemblies so that you can view them in a structure viewer outside. We also have an API here at PDBE. Before I move on to that, we'll talk about how you can find out more about Fold and PISA. If you go to our training tab and click on tutorials, we have a number of tutorials here, and you will find our webinar here once it's been finished. There's a link here for PDBEFOLD, and there's also a link for PDBE-PISA, and these are tutorials to help you find out how to use these services. We have an API here at the PDBE for use by programmatical databases, programmatical access to the data. This link here will take you through to our API pages. And there are some links here to give you more information, for instance, some FAQs and some worked out examples. If you go to our documentation link, this will give you a list of calls that you can run on PDB entry data. If you run any of these calls, this will then return a script for you to use in your bioinformatics work. Before I move on to talk about the EMDB, I'll just give you another quick poll for you to fill out. I'll show you the results here. So there's a very broad range of what people think, and actually the lowest result here is 402, which is actually the correct result. So EM is now becoming a lot better in terms of the quality of the maps and how many angstroms you can get to. Actually, the final answer there, 3,646, is in fact the total in the database. So we're getting more and more each time now. The PDBE is the archive keeper for the Electron Microscopy Data Bank. I'll show you more about how you would find electron microscopy data at the PDBE. So if you go to our website at pdbe.org, you'll see there's a link at the top right here for the EM search. This will take you straight through to search for electron microscopy data. There's also a link for the EM resources, which will take you through to the EMDB pages. You can also find these at pdbe.org/EMDB. There's a few quick links here for high-resolution structures, for instance, or single-particle or tomography entries. If you want to search for a specific entry, you can put this code in here and bring up the entry summary or any visual analysis, and also look at our volume viewer. If you go to the left of the page, there is a link here for search, and this brings up our advanced EMDB search. So you have some ID code searching at the top and resolution searches. You also have some more detailed searches about the samples and maybe about the methods. So you could search, for instance, for single particle entries or tomography entries. Once you've decided on your search parameters, you can then click Submit. There's a number of other parameters you can refine at the top, and your entries are listed down here with some other details regarding the entry. If you go to the entry summary, this will bring up the page for that entry. This gives you an image of the map. You also have the experimental technique given along with the resolution, and some other related entries, either in EM entries or any related models in the PDB. You also get some information further down the page about biological information or experimental information for that entry. The EMDB pages at PDBE have a number of integrated tools in order to help you view an EM entry. And I'll show you how these tools work. So this entry is EMD 3061, and on the right you'll see there are links here for a volume viewer, volume slicer, and visual analysis. If we first click on the volume viewer, this will bring up our volume viewer in a box here. This entry has a PDB model associated with it, shown in coloured ribbons. There's also the EM map shown in an orange colour here. You can also adjust the contour levels of this EM map to give you more or less density. You're also able to change the opacity of the map to see it more clearly, or to maybe see the model more clearly instead. There are of course other viewing controls to zoom, and you can also view a different depth. This might be useful for big entries where there's a lot of other parts of the map in the way. You can clear these away to just see the parts that you're interested in looking at. Another tool we have at the EMDB is a volume slicer. This takes a slice through the entry in 3 dimensions here, through the map. And you'll see in the viewer you have a large image in the middle, along with 3 different views to select from. You have a top view where you can select the position of the image. This can be zoomed in. You also have a front view and a right view where you can take different slices through the map and see the images associated with this. And if you progressively work through this, you can see each layer represented by a single image. We also have a visual analysis section. This includes orthogonal projections, that's projections through the map. There's also a central slice section which gives you images from 3 views through the center slice, and orthogonal surface views which shows you 3 different views of the surface. EMPIAR is the Electron Microscopy Pilot Image Archive, and it provides raw image data to help aid methods development and validation within the EM community. So you can access EMPIAR from the PDBE website via the link on the right. This will take you through to the Pilot Image Archive. You have a number of data sets shown in here. You can also deposit data using the link at the top. These are very large data sets, so we're able to store what would be very difficult to store image sets on our servers. If you link through to one of these entries, you get some information about which images are contained within the entry, along with an image of the map. And further down the page, you'll get image sets, which you can then download. So that's the end of this PDBE webinar. I'd like to thank you for listening and ask if you have any questions, then please send these through the chat box and we'll hopefully come back to you and answer these very shortly. Thank you.